Okay, so this is review for chapter 4. I have to stress again, like I always do, is don't use this exclusively. Go to your notes, your book. Go to the lectures that are on Blackboard. This is just a hitting the high points. Okay. So, cell is the smallest unit that shows the properties of life. Very important. We talked about this before. Hit it again. There are two types of cells: prokaryotic cells and eukaryotic cells. Prokaryotic cells are the archaeans and the bacteria. Eukaryotic cells are everything else: plants, fungus, animals. The point of differentiation is that prokaryotic cells lack a nucleus. By definition, before the core or before the nucleus is prokaryotic. Okay. Eukaryotic cells have a nucleus, and a nucleus, again, as we talked about before, is an area that stores the DNA. All cells, no matter what cell, bacteria, plant, animal, you name it, they're all going to have three characteristics. There's no variation here. One, they're going to have some type of a plasma membrane. Okay, think of this as like an orange peel around an orange. Two, they're going to have a DNA-containing region. Now, bacteria have a different type of DNA-containing region than, say, an animal cell, but we both have an area that holds our DNA, that holds our genome. A cytoplasm. We're all going to have like a juice, in a sense, in the center of the cell. We talk about this later. But we all have it. Bacteria have it, plants have it, and animal cells have it. Now, how we organize that cytoplasm is going to be a little bit different. For example, a plant cell is going to have a vacuole in the center, whereas an animal cell will not. So different ways of organizing the cytoplasm. But the point is what? We all have some form of cytoplasm in our cells, be it bacterial, plant, or animal. So we ask the question, do cells get bigger with bigger animals? And are bacteria not as big as people? Well, the answer to this is no. Cells are the same size for the same function. Which means if I was to look at pancreas cells in a lion and then compare those to pancreas cells in a hamster, they're about the same size. Okay? And are bacteria as big as people? No, they're not. Okay, so what's the point here, and why is that? Well, it turns out that there are limiting factors on how large a cell can get. Okay? And that's why bigger organisms are bigger, because there's more cells and the cells are not larger. This also explains why bacteria are not, very large. Okay? And it all has to do with this concept of changes in volume versus surface area. So cells get bigger, their exterior becomes smaller compared to the room inside, or the cell volume. So what does this mean? This means that the surface area will grow less than the volume inside the cell as the cell becomes larger. And this is important because the surface is almost like the face of an organism, how it brings everything in and everything out. And if the surface is not large enough to service the interior, the cell will die. For example, think of a normal person the height of 50 feet. Okay, really huge person, 50 feet high. But what if their mouth was the size of a normal human mouth? Would that work? No, it wouldn't, because that small mouth could not bring enough food in. It could not bring enough air in to service a 50-foot tall person. Well, the same thing applies here. Okay, that the surface area, this area around a cell, has to bring things in and take things out. And if it doesn't have enough area to do that, it cannot service the interior of the cell. Okay, so this means that cells tend to be very, very, very small. Why? To maximize the exterior volume and minimize the interior volume to the exterior volume. So that's why cells are very small. Okay, it maximizes the exterior surface area to the surface volume inside. So now we talked about lots of different organelles. We talked about the first one is the plasma membrane. Think of the plasma membrane as an orange peel, or the skin around an orange. This is the barrier between a cell and its outside world. So it's the face of the cell, right? Now, I've used this analogy, and I want to make sure we get this analogy, is one function of it is the face of the cell, right? Our faces do what? Well, one, we get information from the outside, right? We hear things, we see things, so we get information from the outside. We take in things from the outside. We breathe in air. What else do we do with our faces? Well, we breathe out carbon dioxide, right? We breathe out some waste. Also, the shape of our face kind of identifies us, right? Okay, well, that same idea applies to the plasma membrane. So instead of eyes and ears, the plasma membrane has receptors on it. These can pick up hormones, neurotransmitters in some cases, but it's going to tell the cell inside things about the outside. Another thing it is, it's for identification. The shape of our face identifies us, right? Well, the cell has things called MHC markers on an exterior. These are going to help identify the cell, okay, as a self or non-self cell. So the point is this: the plasma membrane brings things in and takes things out. And it can also be the location of identification for a cell. Okay. Another function is the gatekeeper. That's my own terminology for it. Essentially, what does it mean? It means it's going to control what comes into the cell and what releases from the cell. Okay? For example, the cell wants to keep some things inside of it. It wants to keep organelles in there. It wants to keep some ions in there. Also wants to keep some things out. It wants to keep bacteria out. It wants to keep viruses out. It wants to keep some ions out. Okay? You need to move things out of your cell. Again, the plasma membrane. It's going to control what leaves, and it controls what enters. Okay? The plasma membrane is made up of phospholipids. We talked about these with the macromolecules. I told you you'd see them again. Well, we're going to see them again. So again, we have a bilayer membrane, and they're made of phospholipids. Bilayer means there's two layers. Okay? A layer facing the outside of the cell and a layer facing the inside of the cell. These plasma membranes, the phospholipids are hydrophobic and hydrophilic. And it's this behavior that makes that bilayer. So one phospholipid has a hydrophilic head, hydrophobic tail. Hydrophilic will want to associate with water. The hydrophobic tail will want to be away from water. Okay, and in the plasma membrane, the hydrophilic heads are faced away from the cell and toward the cell near the liquid. The hydrophobic tails point away. Okay. We use this to kind of describe what happens. In this little video, we have the orange, which is hydrophilic heads, and the gray, which is hydrophobic tails. It's in a mix of water. Okay, and what's going to happen here? Two things. The hydrophilic heads are going to arrange in such a way that they are touching water. Okay, they love water, hydrophilic. The gray tails are hydrophobic. They're going to arrange in such a way that keeps them away from the water. And what do we see? We see that a bilayer has formed. Starting from the outside red ring, we have hydrophilic heads. Moving in, the gray, those are hydrophilic tails. We go to the next layer, hydrophobic tails. And then we have red, which is hydrophilic heads. So again, hydrophilic heads, hydrophobic tails, hydrophilic heads. Okay, the red is hydrophilic heads, the gray is hydrophobic tails. Okay. Talked about different microscopes. We said that what? We talked about the different microscopes, and we said that different microscopes give us different images of the same molecule. And we pick our microscope based on the size we need to see and other conditions if we want to kill the cell or keep it alive. We talked about this in class. Okay. Okay, now here we're coming back to prokaryotic cells, and we talked about these before. Okay. Again, prokaryotic cells are bacteria and archaeans, or archaea, and these both lack a nucleus. Features about a prokaryotic cell. One, it has a cell wall surrounded by a plasma membrane. Okay, so it has a plasma membrane, and then around that it has a cell wall. Some have a flagellum for movement, not all, but some do, and many of them have pili, which helps them adhere to surfaces and can be a mode of exchange of genetic material. The Archaeans, these guys are extremophiles. We talked about several examples in class, but what was the bottom line on it? These are the extremophiles. Okay, they can survive in incredibly diverse conditions. Nuclear reactors, volcanoes or near volcanoes, hydrothermal vents on the bottom of the ocean. They can survive in extreme conditions. Now, it just turns out that bacteria do not live alone, typically. Typically, when you find bacteria, you're going to find it associated with other bacteria, and you're going to find them associated, well, many times, with other forms of life, such as fungus, algae, protozoans. Okay, eukaryotic cells. What's the difference between prokaryotic and eukaryotic? Well, obviously it's going to be a nucleus. And eukaryotic cells have organelles. Organelles are packets inside the cell that carry out specialized functions. Okay, the table on the right are a series of those organelles, and we're going to talk about those organelles as we go through this review. Okay. Here's a plant cell, animal cell, nucleus, a couple things about the nucleus, and now we're talking about organelles. So the next series of slides will be exclusively about organelles. So the nucleus is what? It's going to hold the DNA. Okay? DNA does not leave the nucleus. Okay, what leaves is RNA, which is a copy of DNA, but DNA does not leave the nucleus. In us, human cells, the nucleus will contain chromosomes, which is our genome. So we'll talk about that in a second. Okay. It has a nuclear envelope. Okay, chromosomes. Here's what I want to get to. So a chromosome is a single DNA molecule with all of its attached proteins. Okay, we have 46 chromosomes. Most of us do. In some rare instances, you can have one more or one less. Example of this would be Down syndrome. You have 3 of the 21st chromosome. But typically, most people are going to have 46 chromosomes. Okay. Chromatin, in 46 chromosomes, half of those chromosomes come from your mother, half of those chromosomes come from your father. Now, chromatin is all of the chromosomes and all the protein in one. So the sum total of the chromosomes in a cell is the chromatin. Now, chromosomes come in different forms based on the need of the cell. Sometimes chromosomes come in an uncondensed, opened up form. Think of this as a thing of spaghetti. Okay? What is this good for? And this is the image on the far left. This is good when the cell needs to, quote unquote, see the DNA. Okay, if it needs to access that DNA to make RNA, this form is really good. But sometimes the cell doesn't need to see DNA, it needs to duplicate. And the image on the left, moving this around is like herding cats in a sense. It's not very easy to move around. We can't do something called segregation. We can't move this stuff around. So what we have to do is we have to pack it up really tight or condense it in order to make it into a form we can move. And that's the images in the middle and on the right. So when are you going to see this? You see condensed chromosomes, the image on the right, when you're doing cell duplication, cell replication. because you're going to replicate the DNA, and it's going to be split between two cells. So you're going to need to move this around. Condensed chromosomes, the image on the right, is the best for this. Sometimes a cell does not care about moving DNA around. It's not replicating. It just needs to see the DNA to use it. That's when the image on the left is good, when it's like a unwound piece of spaghetti. Okay? Okay, endoplasmic reticulum, another organelle. Two types of ER we're going to look at. We're going to look at smooth endoplasmic reticulum and rough endoplasmic reticulum. Okay. Rough endoplasmic reticulum, it's rough because it has ribosomes on the outside. And this is where protein synthesis happens. Rough ER is where protein synthesis happens. Before, we talked about in class about what comes out of the nucleus, one thing, that's RNA. Well, a lot of these RNA molecules are going to go to the rough endoplasmic reticulum. Not all of them, but many of them. They're going to go to the rough endoplasmic reticulum, and it will make protein inside the rough endoplasmic reticulum. Okay, the smooth endoplasmic reticulum, this is more for lipid synthesis. It's smooth because it does not have ribosomes on it. So two things that smooth ER does is lipid synthesis, and also it's going to be involved in detoxification. Now, we talked about this in class about the smooth endoplasmic reticulum and detoxification. The more people abuse drugs or alcohol, the better able their smooth ER is able to detoxify. So that's why you get less of a buzz from these things, because you're getting better at getting rid of them. Okay, vesicles. Vesicles are like little balloons, okay, and they're floating around your cell, and they are going to be responsible for the storage and transportation of substances. Examples of vesicles are peroxisomes and vacuoles. Lysosomes, these have digestive enzymes in them. Okay. These are going to fuse with other vesicles and break down what's in the center. Golgi body. Golgi body is essentially the shipping center for the cell. Okay, this is what's responsible for packaging what comes from the factory, such as a smooth endoplasmic reticulum, and packaging it into doing some final preparations to it, and then packaging it for release. Okay, good analogy is an idea of a factory versus a shipping center. Different parts of the cell are the factory, such as the rough endoplasmic reticulum. But once they've made their product, they don't ship it out. Okay, you need to have that. You need to have some final preparations done for that product, and that's what the Golgi body does. Okay, so first it's going to receive and modify molecules such as protein, and then it will then package that, do some final preparations, and release it. Okay, so again, we use this. So first, just an example of how the Golgi body works. On the left, we have the rough endoplasmic reticulum. In the middle, we have the Golgi body. And then on the right, we have the exterior of the cell. So proteins are released from the Golgi body as one. They go to the Golgi body. They fuse with the Golgi body, entering into the Golgi body. Inside, they're processed. These proteins are processed. That's step number 4. Things are added, sugars can be added, shapes can be altered, fine little spit and polish, in a sense, is done to it. Finally, we got some final sorting. 6, they're put into vesicles. Okay, and again, remember we said vesicles are little sacs, right? Well, the proteins are put in the vesicles, and then they are released. Now, a couple different types of vesicles. One type of vesicle, number 7, is it can take that product and release it to the outside of the cell. Okay, hormones release can follow this pathway. Another option for the proteins is that those proteins may be needed on the cell membrane. So it's going to put those proteins on the vesicle, and when that fuses with the outside, it will integrate those proteins into the outer surface. Another option is it's going to take those proteins and go somewhere else in the cell. For example, lysosomes. But the point is what? This thing's going to receive proteins, it's going to package them, do some modifications, and then release them into the cell. We talked about Tay Sachs as being when lysosomes do not work. Mitochondria. Okay, mitochondria is the quote-unquote powerhouse of the cell. Now, as you see, it's the site of oxidative phosphorylation. Don't want to get too much into this now because in the next few chapters we are going to discuss oxidative phosphorylation. But for now, let's just say the mitochondria is an energy converter. What do I mean by that? Well, glucose, here's the analogy we use in class. Glucose is like a $100 bill. ATP is like a $1 bill. Okay? A $100 bill is good for, very compact energy storage, but it's not very useful in a lot of places because a lot of places will not change a $100 bill. Okay, so that's like glucose. ATP is like a $1 bill. It can be used in different places. Vending machines, just about every place you go will take $1 bills. Okay, well, an analogous situation is the difference between glucose and ATP. Glucose is like a $100 bill. Nice, compact form of energy, but it's not usable many places. ATP is like a $1 bill. Very small packet of energy that's usable in many different locations. Well, the mitochondria is going to do that conversion. Okay? It's going to make a lot of $1 bills, or ATP, ultimately from glucose. Now, the specifics of all that comes in later chapters, but for now we're going to say that the mitochondria is the powerhouse, the site of oxidative phosphorylation for the cell. So it's going to use glucose and oxygen and make ATP. And that's the analogy I talked about before. So why do you need this? Where are you going to find the mitochondria? You're going to find it where the cell needs a lot of energy. Okay, you're going to find mitochondria in all cells, but more energy the cell needs, the more mitochondria you're going to find. This one again has that dual bilayer membrane, so it has a two-membrane structure. Mitochondria, how does it replicate? Where is it from? Mitochondria undergo self-replication. They replicate on their own. Where do we think the mitochondria is from? We talked about that endosymbiosis theorem. We think, lines of evidence seem to indicate this, that as the mitochondria originates from a bacterium entering into the cell. Okay, so that's what we think the mitochondria is from. That a bacterium entered into a cell, and over time, that bacterium took up residence and became part of the cell. Lots of things lead us to believe this. It has ribosomes like a bacterium. It's about the same size as a bacterium. It has circular DNA like a bacterium, and it replicates independently. So that's why we think that is really the origin of this organelle. Okay, we also talked about these things too, but I want to get to cell junctions, important point. Cell junctions are the connection point between cells. Okay, we talked that cells make up tissue, right? Well, how do cells connect to each other to make tissue? There has to be some type of mechanism by which cells connect to each other, and that's where cell junctions come into play. We have three types of cell junctions we talked about in class. We talked about tight junctions, adhering junctions, and gap junctions. Tight junctions, those are like waterproof seals between two cells. Adhering junctions, like spot welds. These are very physically strong. Okay, so cells do not separate. Gap junctions. These are holes, channels by which two cells can communicate with each other. Okay, so these are cell junctions. These are things that we find connecting or between two cells. Different type of junction for different types of functions. Tight junction, waterproof. Adhering junction, physical spot weld, connecting point. Gap junction, holes, channels between two cells for the dissemination of information. Okay? Also talked about the cytoskeleton. Now, before we showed a picture in class about the cell, and in class I believe I said something like, this doesn't show the whole story, right? There's a lot more going on here. And one thing that a lot of images do not show is a cytoskeleton. Inside of our cell, there's a highway, a network of proteinaceous filaments, protein filaments, all throughout the cell. Three types of cytoskeleton are microfilaments, intermediate filaments, and microtubules. Microfilaments are the smallest of the three. These are made up of actin. They're involved in cell movement, mechanical support, and when we talk about muscles, we're going to see this come back. Okay, so microfilaments are made of actin. Microfilaments polymerize spontaneously. So each of those little spheres are actin molecules, and those actin molecules are coming together. Okay, and that's what we mean by self-polymerization. Talked about intermediate filaments in class. We talked about those points. You can read the points yourself. And then we also talked about microtubules. These are made up of a molecule called tubulin, and these assemble or disassemble at the ends. These are literally small tubes in the cell. Okay? Here is polymerization, and this is the depolymerization. They come apart and they assemble at the ends. Okay, before we saw polymerization, the tubulin was adding to an end. Depolymerization is breaking apart from the end. So where you're going to see microtubules, major places you're going to see it. Separation of DNA in mitosis and meiosis. Okay? The drug Taxol interferes with that. Separation of DNA, and we'll talk about this with DNA replication in a few chapters. Preview of coming attractions though. The microtubules are involved in the separation of DNA during DNA replication. If you've ever known anyone with cancer, in particular breast cancer, a common drug for this is Taxol. Taxol will interfere with microtubule depolymerization and will actually be a mechanism by which this cancer is treated. So if you ever want to look up the drug Taxol, you can find out more about that. But it's actually affecting these microtubules. Another thing we see in microtubules is it's a pathway for vesicle transport. So right here, that blue sphere is a vesicle. You know, that could be from the rough and or the Golgi body, for example. Okay, and it will be directed to locations in the cell along this highway of microtubules. Okay, so the big purple sphere is the vesicle, and it can be filled with proteins, enzymes, whatever. And really, that doesn't just go in the cell and float around where it wants to. Okay, there's actually a direction to it, and that directivity is done by the cytoskeleton. The cytoskeleton is like a road, and that road will carry the vesicle to its final destination. Another thing that the cytoskeleton does, it's involved in the movement of organelles. Here's a mitochondria. Okay, and it will help move the mitochondria to different regions of the cell where it's needed. Okay, these organelles are not fixed like houses. There's some dynamicness to them. They can move around. Okay, and this is that last part about Taxol. Again, this is a drug that inhibits the depolymerization of microtubules, and it's used in the treatment of cancer. Cancer, again, is what? Uncontrolled cell replication. When cells replicate, they need to take that DNA and copy it and separate it between two cells. A key part in that function is microtubules because they pull apart the DNA and are involved in DNA replication. So if we can inhibit microtubules from depolymerizing, we can, inhibit DNA replication. We inhibit DNA replication, we can treat. That can be a treatment for cancer. And again, that's what Taxol does. Okay. We'll talk more about this when we talk about DNA replication. There's a lot more to talk about here, and it's a lot more appropriate when we're talking about DNA replication. And that concludes this chapter.